Hello everyone, today I'll be introducing you to CRISPR-Cas9, a revolutionary gene editing technology that is transforming the modern medicine. We will explore on how it works, a real clinical case, and discuss some important ethical questions for the future. So, what is CRISPR-Cas9?
These are the definition of CRISPR-Cas9, which CRISPR clustered regularly interspaced short palindromic repeats, Cas9 CRISPR-associated protein 9, and gRNA, which is guide RNA.
CRISPR-Cas9 is a tool that scientists borrow from bacteria. Think of it like a pair of genetic scissors that can find and cut specific sections of DNA. Originally, bacteria used this system to defend themselves against viruses. Now, scientists use it to precisely change DNA in plants, animals, and even humans. So, how does CRISPR-Cas9 work? Let's break it down the process in simple terms, which are four. First, adaptation, and then the expression, and then interference, and lastly, repair. For the adaptation, when a virus attacks, the bacteria save a small piece of its DNA as a memory. And then for the expression, the bacteria make a guide RNA from this memory, which will search for the same virus if it returns. And then the interference, the Cas9 protein uses this guide RNA to find and cut the matching DNA like a GPS-guided pair of scissors. And lastly, repair. In gene editing, scientists use this system to cut DNA at a chosen spot, while the cell then repairs the break, allowing us to fix, remove, or add genes. And then fast forward, what are the current therapeutic applications of CRISPR-Cas9? Since the discovery of CRISPR-Cas9 in 2012, it has been widely used for clinical and therapeutic applications. Some of it are cancer, genetic, cardiovascular disease, and infectious disease research. For the genetic disease, the example chosen here was the sickle cell disease or SCD. CRISPR-Cas9 disabled the BCL11A gene in the bone marrow cells, which boosting fetal hemoglobin and alleviating the SCD symptom. This approach in the clinical trials and represent a breakthrough for inherited blood disorders. As for the COVID-19 research, the rapid diagnostic of the CRISPR-Cas9 tools, including the Cas, enable and fast accurate detection of SARS-CoV-2, which complementing the conventional diagnostic method. For the cardiovascular disease, in the gene correction, they are used to correct mutation in gene like LDR in familial hypercholesterolemia and to model cardiac diseases for drug development. And also for the cancer therapy, there are cancer model and targeted therapy, which the CRISPR-Cas9 is used to create a cancer model, study drug resistance, and validate a gene target. For example, target diffusion oncogene in cancer cells, which can lead to cell death, offering a new hope for cancer treatment. And then we shouldn't forget about the clinical case study of this technology. One of the amazing examples is the treatment of the sickle cell disease, which is a painful inherited blood disorder. In the recent clinical trials, doctors used CRISPR-Cas9 to edit the DNA in the patient own blood stem cells. By turning on the production of healthy hemoglobin, they were able to relieve the patient's symptoms, offering hope for a long-term cure. In fact, the first CRISPR-based medicine for sickle cell disease, called Casavy, was approved in 2023. Besides, what are the advantages of CRISPR-Cas9 over other tools? CRISPR-Cas9 stands out because it is a faster, cheaper, and much more precise than other gene editing tools like zinc finger nuclease, or short form is ZFNs, and transcription activator like effector nuclease, or the short form is TALENs. So instead of designing a new protein for each DNA target, you can just change the guide RNA. This makes gene editing much more accessible and efficient. However, we must not forget about the challenges and ethics. CRISPR is not perfect. There are concerns about the off-target effect where the wrong part of the DNA might get changed. And then there are also the issue of editing genes in embryo or future generations, which raise big ethical questions. Should we be able to change traits that can be passed down? There are also debates about who owns this technology and how to make sure it is used safely and fairly. So, for the future of this technology, CRISPR, if you're looking ahead, CRISPR-Cas9 could help us cure more genetic diseases, develop new cancer treatment, and even create a rapid diagnostic test for infections like COVID-19. With continued research and careful regulation, the possibilities are truly exciting. So before we end, here is a question to think about. If we have the power to edit human genes, where should we draw the line between treating disease and enhancing traits? Who should decide?